Welcome to part 2 of chapter 3, Cell Biology. In this video, we will discuss how substances are transported across the plasma membrane. We will define selectively permeable, discuss 3 passive processes, 2 active processes, and vesicular transport. Let's get started. The plasma membrane is considered selectively permeable. This means that only certain substances are allowed to pass through it and into the cell. Small, lipid-soluble, and uncharged molecules can slide right through the plasma membrane's phospholipids. Large, non-lipid-soluble molecules and ions need transport proteins or vesicles to enter the cell. The plasma membrane's selective permeability is one reason the cell's ionic environment is maintained. It is very important that you know what the cell's ionic environment is. It is having higher sodium ion and chloride ions outside of the cell than inside of the cell. And the cells have higher potassium ions concentrations inside of the cell than outside of the cell. Additionally, the plasma membrane properties maintain the cell's volume even when materials move in and out of it. There are 3 ways to cross the cell membrane. First are passive processes that do not require ATP. They use concentration gradients. In nature, ions want to have equal concentrations on both sides of a membrane. When there's a difference in the concentration, molecules will want to move from the higher concentrated area to the lower, equaling out the concentrations. This is shown in the small image on the left. The 3 types of passive processes we will discuss are simple diffusion, facilitated diffusion, and osmosis. The second way to cross the plasma membrane is through an active process. These require ATP to provide energy because active processes move molecules against the concentration gradient, which, as we just explained, is not what wants to happen in nature. Primary and secondary transport are examples of active processes. Lastly, substances can use vesicles and ATP in vesicular transport. Let's discuss all of these methods in more detail, beginning with the passive processes. Remember, passive processes do not require ATP to move substances across the plasma membrane. In simple diffusion, substances freely cross the plasma membrane, moving down the concentration gradient. This happens with small, uncharged, lipid-soluble molecules moving from an area of high concentration to low concentration in an effort to even out the concentrations. How quickly substances move through a membrane is affected by a few conditions. Diffusion occurs faster when there is a larger concentration gradient, a higher temperature, smaller particle sizes, and less viscous solvents. Viscosity is how thick a liquid is, so less viscous means it is a thinner, more watery liquid. The second passive process is osmosis. Osmosis is the diffusion of water, or a solvent, across a selectively permeable membrane. In this situation, water is the solvent or liquid part of the mixture, and the solutes are particles that cannot cross the membrane. There is a concentration difference between the 2 sides of the membrane, but since the solutes can't cross the membrane to even out the concentrations, the water moves. When the water moves from an area of low solute concentration to an area of high solute concentration, the solutes spread out and the concentrations equal out. This process is shown in the image. The left image shows a high concentration of solutes in the right arm of the tube. The yellow solutes naturally want to even out the two sides, but they're unable to cross the membrane. Osmosis occurs as a result. The right image shows water crossing to the right arm, spreading out those yellow solutes as much as possible. As you may be asking, how does water cross the plasma membrane when it's made mainly of phospholipids with hydrophobic tails? Wouldn't water be repelled? Well, water travels in and out of the cell through channel proteins called aquaporins. Osmosis plays a huge role in healthcare. Consider the administration of IV fluids. The concentration of the IV fluids being administered can alter the patient's blood cells. So it is very important to understand how blood cells react in different solutions. Tonicity is the capability of a solution to modify the volume of cells by altering their water content. Okay, so we can change how big the cells are. Let's consider three situations starting from the leftmost image. Here we see a hypotonic solution. A hypotonic solution is one that has a lower concentration of solutes than what's found inside the cell. So if you see hypo means lower. So there are fewer solutes floating around in that liquid than are inside of that cell. So due to the hypotonic solution, osmosis of water occurs and water moves into the cell, trying to spread out the concentrated solutes. This results in the cell swelling and possibly bursting. The middle image shows a cell in an isotonic solution. Iso means the same. So an isotonic solution has the same solute concentration as what's inside the cell. Here, the amount of water entering and leaving the cell is equal, so the size of the cell does not change. Lastly, on the right, we see a cell in a hypertonic solution. Hyper means above or high, like hyperactive. So a hypertonic solution has a higher concentration of solutes than is what's inside the cell. This results in osmosis where water leaves the cell in an attempt to spread out the solutes in the highly concentrated solution. Because water leaves the cell shrinks, which is known as crenation. So if you think about IV fluids, there are many types of IV fluids that can be administered to a patient. Depending on what's going on in their body, if they're dehydrated, if their cells need to increase in size or they need to decrease in size, the healthcare provider can order different types of solutions. They can be hypotonic, isotonic, or hypertonic. The last passive process is called facilitated diffusion. In this case, there is a concentration difference, but the substance is too big or not lipid soluble, so it can't just flow through the phospholipid bilayer. So a carrier protein must aid the movement of those particles across the membrane. Glucose and amino acids are examples of substances that use facilitated diffusion. Facilitated diffusion is a type of mediated transport. Mediated transport is a general category of transportation in which proteins are used. Pause this video and jot down what you remember about the three types of passive processes we just discussed. Rewind the video to check what you missed. This brain dump activity will help you learn the material. Pause it now. Now it's time to move on to active processes, or methods of transport across the cell membrane that require ATP. The first active process is primary active transport. In this mechanism, substances are moved against the concentration gradient. That is, from an area of low concentration to an area of high concentration. Because of this, ATP is needed to provide the energy to do this. How quickly this process occurs depends on the solute concentration, the number of ATP pumps, and the amount of ATP available. In the image, we see a situation where we want to move the green squares from an area of low concentration to an area of high concentration. The purple protein pump requires the hydrolysis of ATP for energy to change its shape and transport the solute against the concentration gradient. A very important example of this is the sodium-potassium pump. Almost every cell in our body has sodium-potassium pumps. Sodium-potassium pumps are protein pumps that use ATP. Remember when we mentioned that the cell wants to have higher sodium ion concentrations outside the cell than inside the cell, and higher potassium ion concentrations inside the cell than outside the cell. That's how our cells want it to be. In order to maintain that, sodium-potassium pumps are going constantly. The leftmost part of the image shows that there is a concentration gradient for sodium that is higher outside of the cell and for potassium that is higher inside the cell. The sodium-potassium pump is going to pump sodium back outside against its concentration gradient and pump potassium inside against its gradient. Follow along as we go through the steps of the images. First, step 1, sodium from the inside of the cell. 3 of those are going to enter into the pump and bind to it. Also, ATP attaches to the pump. Step 2, ATP goes through hydrolysis to release energy that's used to change the shape of the protein pump, so now it's open to the outside of the cell. The phosphate is still attached to the pump. 3 sodium ions leave the pump. Step 3, 2 extracellular potassium ions enter the pump. This causes the phosphate to be released. Step 4, the release of the phosphate causes the pump to return to its original shape, opened to the inside of the cell. The 2 potassium ions move into the cell. Overall, for every ATP molecule used, the sodium-potassium pump moves 3 sodium ions outside the cell and 2 potassium ions into the cell. The second active process is called secondary active transport. This transport mechanism uses a concentration gradient established during primary active transport to move another substance. An example of this is the movement of glucose into the cell. Once the sodium-potassium pump creates the concentration gradients for sodium being more concentrated outside the cell, an extracellular sodium can enter a transport protein along with glucose. This can be seen in the third protein on the image, so the first yellow one, on the leftmost yellow one. Sodium's concentration gradient pulls it inside, and with it, glucose is brought inside as well. So while the movement of sodium and glucose do not directly require ATP, this process can only happen because the sodium-potassium pump used ATP to create the concentration gradient needed. The third and final active process we're going to discuss is vesicular transport. Vesicular transport moves large substances into or out of a cell by forming a vesicle, and it uses ATP. The video here shows vesicular transport. Watch as the oval-shaped molecule gets enveloped by the cell into what looks like a bubble. That's the vesicle. There are several types of vesicular transport. Endocytosis is the movement of a substance into a cell. If the substance is a solid particle and a large vesicle is formed, it's called phagocytosis. If the substances are small and dissolved and a small vesicle is used, it's called pinocytosis. Exocytosis is movement of a substance out of a cell. Transcytosis is the movement of substances into the cell using endocytosis, through the cell inside of the vesicle, and expelled out of the cell by exocytosis. Let's do another brain dump. Pause this video and jot down what you remember about the 3 types of active processes we just went over. Make sure to rewind the video to check what you missed. Pause it now. In this video, we discussed how substances are transported across the plasma membrane, defined selectively permeable, discussed 3 passive processes, 2 active processes, and vesicular transport.